We now go to the last talk of the session by Ajay Subbaroyan. He'll talk about what appears to drive complex cellular decisions.
Very good evening to all. Firstly, I would like to thank the organizing committee for giving me the opportunity to present my work in front of such an esteemed audience. So the topic of my presentation is simple regulatory logic appears to drive complex cellular decisions. So over the next few slides, I will be explaining how I reach this conclusion. Right. So the first thing is that a cell has takes in many inputs, processes it through its signaling and gene regulatory networks, and this eventually leads to a variety of outputs, which range from cellular differentiation, cell growth, and apoptosis, to name a few of them. Now the question is, how do you mathematically model such a system? So the mathematical framework which I would be focusing on is known as the Boolean modeling framework. Yeah. So I would be explaining what a Boolean model is in this following slide using an example of cell differentiation in pancreatic cells. So the first component of a Boolean network is the network structure, where each of these nodes in this network represents a gene, and the incoming edges to any particular gene represents the regulators of that gene. So each gene takes two expression states in a Boolean network, either 0 or 1. So the gene is either on or off. And so if you take the entire network, you would have, you can represent it as a binary vector, which essentially corresponds to the gene expression pattern of the system. Associated also with each of these nodes is a Boolean expression. So what does the Boolean expression do? It's the sort of the combinatorial regulatory logic at each node, which essentially tells that particular node how to evolve in time. So if you take PTF1A at time t plus 1 is a Boolean function of the three inputs it has. Now, so basically, if you evolve the system, so you have 5 genes, and you have essentially 2 power 5 states, because each gene can take 0 or 1, and so you can have an attractor structure of this kind once you apply the logic rules recursively. And as you see, there are 3 fixed points in the system, and each of these fixed points corresponds to one particular step. Yeah, you can also have cycles, but then this particular model doesn't have cycles. So yeah. So the focus of my work is really on the Boolean functions. A lot of studies have gone into studying the network structure typically of gene regulatory networks and signaling networks, but not much research had gone into what is the nature of logic rules at these nodes. And so that is going to be the focus of my work. Right. So in order to understand better what is the nature of the logic rules, it's important to understand, one can actually look at different representations of logic rules apart from just looking at Boolean expressions. So the first one I'm sure everyone is familiar with is the truth table. So this is the truth table of a 3-input function. And so I denote by k the number of inputs, and the number of ones in the output column of any truth table is going to be called a bias. So this has a bias of 3 because there are 3 ones in the output, and this is also called as, I'll simply call it as an odd biased function. So you can have an even number of ones in the output, that would be called as an even bias. The next one is already something which we have seen, is the Boolean expression, where you have literals and you have, so this is a particular DNF corresponding to this truth table. So each of these clauses is basically the rows corresponding, which have the output value of 1. And this expression can basically be factored to give a much smaller expression like so. The last representation of the Boolean function which would be useful for us is the hypercube. So each of these vertices of this cube is one row of the truth table, and the color associated with the vertex is the output value. So red colored vertices are the input combinations have the output value of 1. So it's easy to see that the total number of Boolean functions for a k input function is 2 power k exponential and exponential in k. So this basically, so if you go to 5 input functions, you essentially have of the order 10 power 9 functions which you could assign. So now it's important then to ask, out of the space of all possible Boolean functions, are there smaller subspaces of Boolean functions which are biologically more meaningful than others? Because then, one doesn't want to explore all possible cases and wants to ask if there are, smaller subsets which are more meaningful. And so that is the first problem that we address, is that what are the possible types of regulatory logics which are more biologically meaningful than others. So I'll illustrate this particular the space of biologically meaningful functions using the space of all 4 input functions, though this is not specifically restricted to 4 input functions. So one can imagine the space of all 4 input functions is this huge square, and then you can divide it equally into functions with even and odd bias. So bias, remember, is the number of ones of the output column of the truth table. Then you have effective functions. So what is an effective function? I'll define that by defining what is an effective input. A Boolean function is said to be, an input x1 here is said to be effective if by keeping x2 and x3 fixed and you flip x1, for some combination of x2 and x3, you will flip the output x4. So that's an effective function. And you can see here that effective functions occupy a huge space of the Boolean functions, whereas there's a small space of functions occupied by the ineffective, and they always lie in the even bias. And this is true for all values of k. Then you have canalizing functions. So an input is said to be canalizing. So the input x1 here, for instance, is said to be canalizing if when it takes the value c1, it fixes the output of the Boolean function to O1, irrespective of what x2 and x3 are. So it is like a veto function in some sense. The next class of function is the unate functions, which is also called as the monotonic functions. So if one focuses on x1 and x4 here, x1 is an activatory function, in the sense that if you increase x1, you either increase x4 or you don't change x4. So you cannot increase x1 and decrease x4 because the relationship is activatory. So that's a unate function. Next is the read once function. In this function, each variable of the Boolean which regulate a particular gene appear exactly once. And you can have any combinations of parentheses in this expression. And the last class of functions we look at are the nested canalizing functions, which is basically an extension of the canalizing functions, in that if the first variable x1 did not take its canalizing input, then another variable, if set to a canalizing to a variable, would fix the output x4. So the take home from this particular figure is really not the shapes. The shapes of this is not important. This is just a Venn diagram made to look fancy. But the important part is at which places do each of these different classes lie, and what is the relative size of the different classes. So you see here that the NCFs are the smallest subspace in the space of all functions here. And after that is the ROFs, then you have the UFs, and then you have the CFs. And you see that also the ROFs and the NCFs always lie in the odd bias, and this is true for all values of number of inputs. Now, this is the theoretical space of all functions, but then the question has to be asked as to are there some types of these functions which are more enriched in real biological data sets? And so we then set out to to collate a set of Boolean functions derived from 88 Boolean models which have been reconstructed till date. And from this 88 Boolean models, we extract all the Boolean functions and study them. So what is the first observation we make from this is that you can compute the in-degree distribution of this set of 2687 functions. And the first thing you observe is that most of them are odd biased functions. That is, the number of ones in the output column of the truth table for each of these functions is an odd number, which is actually surprising. If functions were chosen randomly, then you would expect that you would have an equal number of evens and odds. but this is otherwise. Then we look at how each of these different classes which I mentioned to you, that is the effective unit canalizing ROFs, NCFs, and non-NCF ROFs, how are these enriched in the real data set? And we find that indeed the ROFs and NCFs are highly enriched. And in fact, that they are also relatively enriched, meaning that not only are they enriched in the space of all functions, but NCFs being a subset, say, of the CFs and UFs are also enriched within that smaller subspace. Right. And so we've answered the second question. Then, are there some classes which are more enriched than others? Yes, NCFs and ROFs. Then the question is, what properties of these types of Boolean functions can explain this enrichment? So we perhaps thought that one could look at the complexity of these functions and then to explain. But then, how do you define the complexity? Is there a quantitative way to do so? And so we resorted to the computer science literature to borrow some measures from there and apply them over here. So the two measures of complexity we look at are the Boolean complexity and the average sensitivity. So what is the Boolean complexity? It is the number of literals in the shortest formula equivalent to some given formula. So for instance, you consider the following expression. One can actually apply the laws of Boolean algebra and reduce it to a smaller expression here. Of course, and you just essentially count the number of literals in this expression. So here you have 5. X2 appears both in its negated and non-negated form. So you count both anyway. So the Boolean complexity of this expression would be called as 5. So if you take all 4 input functions, 65,536, and then you plot the Boolean complexity versus the bias, this is the plot that you would get. I'll mention here, there's a big caveat, which is that these are not exact computations because, of course, this is a computationally intractable problem, computation of Boolean complexity. So this is only approximate. But what is true here absolutely is that the NCFs and the ROFs would have the minimum value. That is, this is in some sense the measure of compressibility of a function, and you cannot compress the NCFs and ROFs further than this. So the ROFs and the NCFs basically have minimum Boolean complexity. Yeah, the next quantity is we look at. So, but if you notice, this doesn't distinguish the NCFs and the non-NCF ROFs. And so we needed to look at some other measure of complexity, and we look at the average sensitivity. So what is the average sensitivity? For that, we need to define what is the sensitivity. Sensitivity is defined at any vertex of the hypercube, which is a row of the truth table. So the sensitivity of the vertex 0, 0, 0 is basically the number of neighbors which are different from itself. So 0, 0, 0 has only one neighbor which is different from it in terms of its output value. So the sensitivity of 0, 0, 0 is 1. Similarly, one can compute the sensitivity of all the other vertices, and you can get the total sensitivity and the average sensitivity, which is basically the average over the total number of vertices. And so then what we did is that this is an exact computation, so there is no problem here. And so one can essentially do a calculation of all the 65,536 functions again and see how these different types of Boolean functions, where do they lie in this. Very interestingly, if you notice that at each of these odd biased values, you can look at all these triangles, these are the NCFs. And whereas the ROFs are rectangles, squares, and they are sort of above the NCFs. And so this measure sort of distinguishes the ROF and the NCF, or rather the non-NCF ROF. And so then this made us to really conjecture, this led us to conjecture that the NCFs have the minimum average sensitivity for a given number of inputs and for any odd bias. And so the last slide is really to show that this is indeed the case, that there exists a theoretical proof that one can show that for any value of k and for any odd bias, the NCFs actually minimize this average sensitivity. And so it basically amounts to finding some arrangement of these p vertices such that you will minimize the average sensitivity. But this problem can be transposed to something else, which is basically counting the number of edges between the vertices which have output value of 0 and the output value of 1, and trying to minimize those number of edges basically. But this particular problem we found had been solved in 1976 by Sergio Hart, and in this particular paper called A Note on the Edges of the N Cube. And he had solved this problem basically, and because that solution was available, all we had to do was to show that the solution he provided was equivalent to an NCF. And that's what we really show. And so his solution is basically called a good set of vertices. So what is a good set of vertices? So if you have, if you want to look for p vertices on the hypercube, which would minimize this average sensitivity, you essentially have, you will say that p lies between some 2 power r and 2 power r plus 1. And then you would choose this an r plus 1 dimensional cube, break it into 2 vertex disjoint subsets of r cubes, and fill one of those with p vertices. And then you repeat this process recursively on the other r cube. And so, so that's really the result. And so finally, what are the properties or features of Boolean functions that could explain the enrichment of ROFs and NCFs in the empirical data set is the minimum Boolean complexity and the minimum average sensitivity. And so therefore, my title, which is that simple regulatory logics appear to drive complex cellular decisions. So there's a reason why I have the word appear here, specifically because the empirical data set which we look at is not free from subjective biases at some level. And so it's important to use the word take a pinch of salt and make the conclusion. And so I would like to thank my PhD advisor, Professor Arijit Samal, and our collaborator, Professor Olivier Martin. And this paper is a published piece of work. And if you would like to have a more detailed discussion and give more criticisms, feedbacks, whatever, you're free to visit my poster, which is also put up. And I would like to end finally by this quote by Leonardo da Vinci, which is that simplicity is the ultimate sophistication. Thank you.